Hello dear students, again I welcome all of you in the second part where we are discussing about life cycle of Plasmodium vivax. As far as part 1 is concerned, we have already discussed about diagenetic nature of Plasmodium vivax, where we discussed asexual cycle and sexual cycle, how it occurs in the human being and mosquito. Today we are going to discuss about whole life cycle of a Plasmodium vivax, and this life cycle which occurs in the human being, that is asexual reproduction occurs in human being, and sexual reproduction or sexual cycle which occurs in mosquito, that we will discuss in detail. So very firstly, as we all know, that entry of a sporozoite which is an infective form which is responsible for causing malaria, when it enters in the body, we all know it travels for half an hour in the body, and then it moves towards very first part of the body, and that part is nothing but a liver. We know that we have divided the cycle, we have divided the stages of this Plasmodium vivax. Very first one is exo-erythrocytic, second one is the erythrocytic phase stage, and third one is the sporogony stage. Now sporogony stage, as we all know, that it occurs in the mosquito. First of all, we are going to discuss in detail about life cycle in human being. Here comes the detailed account how it infects in the human being, how it undergoes very first stage, and that is the exo-erythrocytic stage. We can say that this stage is pre-erythrocytic stage also. See here it is written pre-erythrocytic cycle, means which occurs outside erythrocyte, outside red blood corpuscles. See here, sporozoite, it enters in liver cell. See here it is the sporozoite, it enters in the liver cell. Now after entry into the liver cell, nucleus that grows, it forms a structure which is quite oval in nature, and to this structure we call it as schizont. Now this schizont in the liver cell is formed after entry of this sporozoite. Sporozoite enters, uses liver cell material, food for itself. Then this schizont that divides asexually in the liver cell. That's why this stage is also called as the liver schizogony. Schizogony is nothing but asexual type of division. Now after division of this schizont into many or numerous numbers, they forms number of cryptozoites, and cryptozoites they released, and these released cryptozoites again that invade liver cell, that enters into the liver cell. See here it is cryptozoite entering in the liver cell, another liver cell, and it turns into a schizont exactly similar like a first structure which is formed from the sporozoite. See, we are studying divisions, we are studying cycle, pre-erythrocytic cycle or exo-erythrocytic cycle, which is happening in liver cell. See here it is a schizont formation from cryptozoite. First, what happened? Sporozoite entered into the liver cell, it formed schizont, then that schizont divide asexually to form cryptozoite. Cryptozoites liberated and they infect another liver cell. After entry into the liver cell, it forms a schizont. This schizont again undergoes a sexual development to form numerous numbers of cells, and these are nothing but metacryptozoites. See here, very first one, sporozoite to schizont, schizont to cryptozoite, cryptozoite to schizont, and again schizont to metacryptozoites. Now this is all about a first stage, that is exo-erythrocytic stage of Plasmodium vivax. This formed metacryptozoites now enters in the red blood corpuscles. After damaging to the liver cells and after forming metacryptozoites, it enters into the red blood corpuscle. See here, here is metacryptozoite which enters into the red blood corpuscle, and after entry into RBC, it forms quite oval to rounded structure, and this formed in the red blood corpuscle, it is called as trophozoite. Now this trophozoite undergoes several changes. Here you can see in the trophozoite there is formation of a vacuole, and there is a formation of a ring-like structure. Due to formation of vacuole, there is formation of ring-like structure. That's why this stage in the red blood corpuscle stage is called as signet ring stage. After entry into the RBCs, it is turning into trophozoite. Trophozoite undergoes changes, trophozoite undergoes divisions, and that forms vacuole. Due to vacuole, formation of ring-like structure, and a stage is called as signet ring stage. Now this formed stage undergoes changes. It forms a shapeless type of structure, amoeba type of structure in the red blood corpuscle, and a stage is called as amoeboid stage. This amoeboid stage is changes to again schizont, and there you can find a small granules, and these small granules are called as Scuffner granules. Scuffner granules are formed due to change in structure of amoeboid stage. Now this formed schizont with Scuffner granules, it undergoes a series of division. It undergoes schizogony. It undergoes asexual changes, asexual development, and it forms numerous number of merozoites. And these merozoites again liberated to form a structure which is called as there are two type of structure. One is a larger one, larger merozoite, and quite smaller merozoite, and these structure are called as gametocytes. Due to different shape of these gametocytes, that is smaller and larger, they are named megagametocytes due to larger shape, and microgametocyte due to smaller shape. See, this is a complete asexual development which can be seen in human being. See, first stage is exo-erythrocytic stage, where sporozoite enters into the liver cell. It turns into the schizont. It undergoes asexual divisions to form cryptozoites. These cryptozoites liberates, again invade another liver cell, and these cryptozoite, just like a previous schizont, it forms a schizont structure. This schizont also undergoes schizogony, that is asexual development. It forms metacryptozoite, and metacryptozoite is a form which now start to invade red blood corpuscle, that is RBC, and after entry of metacryptozoite it turns into a first form in the RBC, and that is called as trophozoite. Trophozoite turns into vacuole formation, which gives a signet ring stage, and then this changes to this amoeba like structure, and a stage is called as amoeboid stage. This amoeboid stage changes to a rounded to oval structure, and this schizont that is responsible for the formation of merozoite, and these merozoite they gives rise to gametocytes of two shape. One is larger one and one is a smaller one. Depending on the size, they are called megagametocyte and microgametocyte. The stages which are completed in the red blood corpuscles or asexual divisions which undergone in the red blood corpuscles, that schizogony it is called as erythrocytic schizogony, and the schizogony which is undergone in the liver cell called as liver schizogony. So here comes the end of two first stages, that is stage number 1 liver schizogony, that is exo-erythrocytic schizogony, and second one is nothing but erythrocytic schizogony. Now here comes the end of life cycle in intermediate host. Here comes the end of life cycle in the human being. Now when again there is bite of mosquito, with the bite it sucks the blood, with the blood there comes RBCs, and with the RBCs it takes gametocytes. Now both the gametocytes are engulfed, are sucked by mosquito. Now this goes in the gut of mosquito, and here comes the beginning of a sporogony in the mosquito. Here comes the beginning of a third stage, and that is a sporogony stage which is taken place in the mosquito. Now the development of micro and megagametocyte and changes in the structure of both these gamete that occurs in the mosquito. The changes and the stage that we will see in the part 3 of life cycle of Plasmodium vivax. Thank you.